हेलो। हेलो, माइक चेक। 979 Yeah, I started the. Yeah. So, Amish, how about the YouTube channel? I have done everything that I need to do, right? It is started already. Should we start? But when will it started live streaming? But should we do that? We heard. हेलो, माइक चेक। हेलो। But this video. Hello, everyone. I think we'll start. So, welcome to the Wednesday colloquium. Uh, Jodishman is traveling, so he asked me to fill in for him. So, I'm doing just that. Our speaker today is Mahendra Sonawane, and Mahendra, of course, needs no introduction. But nevertheless, I'll just briefly kind of trace his research trajectory. Mahendra obtained his PhD in 2000 from the Pune University. Uh, After which he went for his postdoctoral research at Max Planck Institute for Developmental Biology in Germany, where he started his the current research interest, his model on zebra fish and on epithelial morphogenesis. He joined TIFR in 2008, and he's a full professor now and also a co-chair of the biology department. So today he will talk to us about cell geometries and patterns on the cell surface. Mahendra.
Thank you, Manoj. Thank you, JD, who's not here, giving me this platform to present some of our recent work. I'm trying to keep things slightly superficial. I'm saying this to biologists. That doesn't mean, as you know, that we don't do deep research. We do thorough things. But for today's audience, I will keep things broader. And if you have very specific questions, we can discuss them later, or maybe in the annual talks, which are going to happen anyway in next 2 to 3 weeks. So I'll start with something very simple, and that is something that we all know, that our bodies are formed by cells. But the point I want to drive is that look at the complexity of the organs that these cells form. And eventually, these cells are going to make up this body. These organs are going to make up this body. So it cannot be that cell geometry will be, identical when you form different kinds of organ systems. For example, this is your small intestine. It's like a tube. This is heart pumping the blood, and look at the complex organization it has. The lens, which is basically an ellipse or ellipsoid body. So you also need to have geometries of the cells accordingly, defined so that you can build such complex organ systems. And this is very obvious from something simple as this building here. If you want to make an arch out of the bricks, you can't use the same bricks that you use to make a building. Look at these shapes are much different than these shapes and sizes. So the same logic basically applies. If you want to build something as complicated as that, you have to manipulate and modulate the geometry of the cells. Or the shape and sizes is what I mean when I say geometry. So where does it all begin? When do these organs form, the tissues form, and how does the animal axis actually forms? It starts with the just stop this video. It starts with the fertilization of the egg. And what fertilization achieves is the division of this one cell, giving rise to balls of cells. No matter what animal you are, you will eventually have some kind of a collection of cells, and these cells will be defined. Their fates will be defined. Some of them will form brain, some of them will form heart, some of them will form skin, some of them will form gut, etc., etc. And eventually, this ball of cell will acquire this form, especially true invertebrates having this kind of, arrangement wherein the organs are organized in a very stereotypic manner along the axis. There are two axes that you will see in any animals. Let's look at the vertebrates. You will have head to tail, this anterior posterior axis, and you will have back and belly axis, which is the dorsal ventral axis. And if you see, the brain is usually on the anterior side of the animal, and the gut is typically in the middle somewhere. And also on the ventral, it's towards your belly, and your vertebral column is on the back side, which is the dorsal. So these two axes, how do they get defined and organized, was the question which was asked long time back. The original research was done in Drosophila as a model, using Drosophila as a model, and the genes which are important for setting up of this anterior, posterior, and dorsoventral axis were identified, and this research was awarded by a Nobel Prize to Ed Lewis, Christian Nusslein-Folhard, and Eric Wischhaus. Now, in principle, what happens is that when you set up such axis, what happens is that mother deposits some proteins or RNAs inside the egg, even before it gets fertilized. And somehow there is some kind of very rudimentary patterning which exists inside the egg. So eggs are not symmetrical. They already come up with some asymmetry. And the fertilization basically sets the cascade of signaling events, which then defines and redefines this patterning, eventually giving rise to head-like structure. So I'm not going to get into the complications of how this whole thing works. It's a very simple schematic here. There are many different kinds of pathways and molecules that are involved. What happens in principle is that there are a bunch of proteins which will not let these cells become, say, for example, tail-like. So these cells will always form head because they will prevent the signal which will give the caudal characteristic or tail-like characteristics. And there are also signals here in the same part which will also prevent these cells from becoming epidermis. They will remain such that they will eventually give rise to the brain. And these are the molecules like BMP and Wnt, which were prevented from entering in these zones by these bunch of proteins. And then some of these mechanisms, of course, there are different kinds of such examples that you will see during early development, which allows the animal to have this dorsoventral and anterior posterior patterning. And this is one important aspect of animal development. How do you get this patterning? But just specifying the cells is not enough. So imagine you are now saying that these cells will form brain, these cells will form heart, but I showed you the morphologies of these organs. They are quite intricate. So these cells basically have to form these organs or these tissues, right? So brain, heart, etc. Now these cells therefore have to kind of undergo some kind of sculpting and form such tissues. So I'm going to talk a little bit about that, where the geometry actually becomes quite relevant. Now this is I'm going to show you two videos. One video is basically of the Xenopus embryo, which is basically going to depict the formation of the neural tube. So neural tube is the precursor of the brain eventually, so to say, in simple words. And the second video is of Drosophila gastrulation, wherein the cells will enter inside the interiors of the embryo, and eventually some of those will form gut cells, and some of those will give rise to muscles and other tissues. So let me play this movie here. Okay. So if you look on top here, what you will see is a thickening of the embryo, and there is a depression here, and this whole thing will go in. And eventually, what you don't see here is that in the end, what you will get is a tube inside. So basically, this is a plate which folds onto itself and then eventually forms a tube which detaches from the overlying tissue, as has been shown here. And I'll come to this in a minute. And something very similar happens in this video, the second video, wherein you will see that these cells are actually going in. And also initially they will form some kind of depression, and of course in the end they will get integrated inside the embryos. And if you look at these schematics here, they depict what exactly happens in these two videos. So initially the whole process starts with something like a sheet here, which is of cells. And some of these cells start basically buckling in. And what provides at least partly is the force that is provided for this whole process is by the change in the cell shapes. You will see that these cells are now narrower. They are wedge shaped as compared to these cells, which were originally more cuboidal. And eventually, of course, this tube will detach from the overlying ectoderm, and then it will give rise to the neural tube, which will eventually differentiate into the brain cells. Something very similar happens here. You will see the changes. If you look at these cells from the top, this is a surface view. You will see the changes in the cell shape. These cells are really constricted, while these cells are elongated. And if you look at these in a section here, you will again see that the central cells are more wedge-shaped, because that's where you are actually having the constriction. If you now play with the machinery that is important for constriction of these cells, then these events, these morphogenetic events are majorly disrupted, giving rise to the phenotypes like open neural tube, to say, as one as an example. Now, what is the machinery that is involved in modulating the cell geometry? Now you have a rectangular cell. Now it's not rectangle, it's in 2D, but I mean, imagine this in 3D happening. Now you have to constrict the apical surface, right? That's what basically you have to do if you want to get this morphology. And there is a machinery that exists on the apical side of these cells. So there is a contractile apparatus which is formed of actin and myosin. So actin is basically, it's a polymer of a protein called actin. So it's filamentous actin, so it forms long polymers. And there is myosin, which is a motor, and this motor walks on these filaments, and it can constrict or bring these filaments together. So this is the contractile apparatus. Now, it cannot act independent of the cell membrane. Eventually, if you have to shrink the cell, you have to connect this contractile apparatus onto the cell, tether it onto the cell, so that when this contractile apparatus constricts, the cell will also constrict. And that's what is achieved by these kind of junctions. These are called adherent junctions. So the cells are connected with each other, not in isolation. And this contractile apparatus is anchored over to these junctions, so that when the constriction happens, the cell membranes are also kind of brought together. And now imagine all these cells constricting at the same time. This would give kind of enough force, in some sense, for the epithelium to buckle and invaginate. So this is basically one major mechanism by which cells can modulate their shapes. Now, the embryos and these developing tissues basically have come up with a variety of different ways by which this contractile apparatus can be regulated in time and space. And there are signaling mechanisms which eventually will impinge on this, so that you can get these modulations in the cell sizes and shapes at the right time during development. Right. So, what I did not say is that most of these morphogenetic events start with this kind of cellular organization, wherein cells are connected with each other tightly. So this organization is typically called as epithelial organization. In contrast to cells which are solitary and can migrate away from each other or loosely connected, those are called mesenchymal cells. So these epithelial cells, now this morphology is not just unique or present during the developmental systems, during development in these morphogenetic processes, but this epithelial organization also exists in the tissues, in the organ systems as well. So some of the organ systems, sorry, this is going to, that have this epithelial organization are depicted here. Your lungs basically have the epithelial, they are formed, their inner lining is formed of the epithelial tissues. Your gut the inner lining of the gut is formed of epithelial tissues. And there are many such examples. These epithelial tissues basically act as barriers. They do not let any foreign particles enter your body. And of course, they are also important for absorption, secretion, and gaseous exchange. And again, if you look at these variety of different epithelial systems, they look at the cell sizes and shapes. So they're quite distinct. I mean, some of these are columnar, some of these are cuboidal, some of these are flat squamous cells. And these morphologies are maintained throughout your life. It's not that tomorrow your gut is going to be formed of cuboidal cells. It will remain, having these columnar cells. So the question then one can ask is, how does the animal or the living systems acquire a particular cell size or cell shape, modulate it when you require them during development especially, or during migration, and how do you maintain them? And these are the kind of questions that we're asking using zebrafish as a model system. And in addition to this, what another feature that the epithelial cells show is that their apical membranes are modified. They have these microvilli-like structures. So this basically finger-like projections of actin. I'll come to this a little later, and they increase your absorptive surface in some sense, so that you can have the larger the surface area, you will have better absorption. So we also look at this, but this I'll come to this part a little later. I'm going to start with this part, how the cell sizes and shapes are acquired, and especially today in today's talk, I'm going to focus on how you maintain the cell size and cell shape. Once you form a particular cell with a particular shape and size, how do you maintain it? So we use zebrafish as a model organism. As many of you know, they can be actually grown in such tanks at very high densities. This fish is actually a very small fish. It's just 3 to 4 centimeter long, and the zebrafish larva is about 2 to 3 millimeter long. And we work on the outer epithelium of this larval fish. And this epithelium basically is formed of 2 layers of the epithelial cells. The outer layer is called as periderm, and the basal layer is, of course, called as basal epidermis. And if you look at these cells from the top, they look like this. They basically, you see this honeycomb pattern. They're polygonal mostly. And here the outer cells are basically labeled using a membrane tethered GFP, and that's why you see this green fluorescence here. And you see that how flat these outer cells are. And on the top surface of these cells, which you see these projections, and if you look at them from the top, that's how they are. They are quite extremely long worm-like projections on the surface. Now, the advantage that zebrafish offers is that it allows it to manipulate the genes, either by doing mutagenesis screens, wherein you use chemicals to inhibit, induce mutations, which would then allow you to do loss of function analysis of some sort. You can also knock out gene, or you may have heard CRISPR-Cas9 system, which can be used for such purposes. And you can also use pharmacological or chemical inhibitors to block a particular signaling pathway or a protein. And there are other cell biological advantages. You can do live and fixed imaging, and this can be done at not just at the tissue and cell level, but you can actually go to the subcellular level. You can image inside the cell what are the events that are happening inside the cells. And you can quantify what is the biggest advantage is that the images are so clean and clear that you can quantify the morphometric parameters of the cells. How big is the apical surface? How big is the basolateral surface? What are the heights? And if there are any changes, one can actually now measure what are the changes. So this is one example wherein we have measured here the entire surface area of the cell, and there are two conditions wherein things don't really change much. Now, where did we start? So why we even got interested in the maintenance of cell size and cell shape? And the reason was isolation of this mutant long time back when I was a postdoc. This is called goose pimples. So the interesting thing about the Drosophila and zebrafish field is that the mutants are named typically based on the phenotypes. So this new mutant was named goose pimples because you see these rounded up cells and they resemble as if the fish is having goosebumps. Eventually what we figured out is that the goose pimple, this gene basically is myosin 5B, and this is a motor which walks inside the cell carrying cargoes. So these cargoes involve basically vesicles which are destined to go towards the membrane. And what happens is that in the absence of this motor, the cells accumulate all this cargo which is destined to go to the membrane. It basically gets accumulated inside the cell, and the cell now has reduced amount of total membrane, and that's why the cells shrink over time and eventually they round up. Now you have to keep in mind that the cytoplasm doesn't shrink. It's only the membrane, the surface area basically of the cell reduces over time, and therefore these cells round up. And that's how we could explain this phenotype here. A quick detour. So the mutations in this gene myosin 5B have been shown to be causal for a disease in humans. It's called microvillus inclusion disease. It is characterized by intractable diarrhea and leading to dehydration and death of the newborns. And if you look at the gut, basically the typical folding that you have in the gut is missing, and the cells basically are devoid of the microvilli, the finger-like projections that I spoke about. And the fish also shows some of these characteristics. You see that the clear absence of the folds, which are there in the wild type or the normal fish, and the mutant fish doesn't have this. And this we kind of published this as a model for this human disease some time back. And it's mind-boggling to see how the mechanisms are conserved all the way from fish to humans. And if somebody questions you about evolution, tell them to talk to biologists. And there are multiple such examples which exist, which convince you that evolution indeed has happened. Right? Coming to this myosin motor again, so eventually what we figured out rather recently is that myosin works in combination with two different adapter proteins, RAB11 and RAB10. And when myosin interacts with RAB11, its function is important for this membrane recycling. And if you abrogate this interaction, the membrane vesicles accumulate, then cells round up. But this interaction of myosin 5B with RAB10 is important for the pinching of the vesicles from Golgi. So this is some kind of a sorting station from where the membrane and membrane proteins are delivered to the plasma membrane. And what happens is that if this interaction is abrogated, these vesicles cannot be pinched off from the Golgi, and they do not reach their destination, basically, which is the apical membrane. And as a consequence, the cells cannot grow. Their apical domain doesn't grow. And that's what we found out. Now, while working on this, the obvious question that came to my mind is that this requires a lot of energy, right, to maintain cell shape and cell size. I mean, why should be so much energy invested? Why should so many proteins, so many, very sophisticated regulations be involved in maintenance of something as simple as cell size? Why can't you now form an animal which will have bigger cells? How does it matter, right? But that's the question that we started asking. Now, if you make the cells big, and if you form the epithelium with bigger cells, would that have any consequence to the animal? And how do we go about this? So what we did is that we inhibited proliferation, and when you inhibit proliferation, the cells actually grow big because your number is now reduced. Cell proliferation basically achieves more number of cells, and if you inhibit that, the cells have to grow bigger so that they can cover the entire surface of the fish. Now you can imagine, you can see that there is obviously the cells are obviously larger, and one can ask this question of, what is the consequence in variety of different contexts, whether there will be physiological consequences, whether there will be any mechanical consequences. And we ask this question in the context of the whether the mechanical properties of this epithelium will be compromised if you form the epithelium with the bigger cells. Yeah.
This is a very physics like. Yeah, so I think it's a naive question. If I imagine that I'm sort of trying to fill out a two-dimensional surface with equilateral triangles, all of the same size. So as long as at every point 6 triangles meet, it makes a plane. Mm-hmm. If I try to crowd around, like 7, 8, etc., then it bends into a convex surface. And if I have less than 6, then it's a concave surface. So this could be one easy way of an animal or an organism to form curved surfaces like convex or concave just by having less or more cells.
Absolutely.
So but of course I have to freeze the size of the cell. So I think here now you're saying that. Yeah, so like if you say that I inhibit proliferation, my tendency would have been to answer that it should make a concave surface.
The inhibition of proliferation doesn't going to make all these cells say 5-sided or 6-sided or 7-sided. So those are not linked things.
I see.
Yeah, in fact, you're right. This is something that Mustansir pointed out to me once. If I'm not wrong, if you have all the pentagons connected together, it has to form a curved surface or a hexagon. I don't know which one.
I was thinking of hexagons or even triangles, which is equivalent. So I see.
No, but there is heterogeneity in that sense. It's not an identical. Some of cells are not identical in that sense. But more or less the sizes and shapes are maintained. And when I say shapes, they do fluctuate. Some of them are pentagonal, some of them are hexagonal. So we typically call them as polygonal. But we don't really see, 4-sided cells. Or sometimes, occasionally 7-sided, but not beyond that. Usually, it is between 5 and 6-sided polygons. Yeah.
Thanks.
Yeah.
The heterogeneity you have, is that something that as the cell grows, it kind of fills in the space around it, or is it, I don't know, pre-programmed in already when the cell is born, it's encoded somehow what shape it will?
I don't think it's encoded. I doubt. It probably depends on how many neighbors it will have around it. I don't think it's encoded. I doubt.
And the contractile apparatus you showed in slide 4 or something, is that there all over the cell, so it can change its shape by squeezing one side, and or is it only on one side and then the cell has
it is typically present on the apical side of the cells, at least in those cases. But I don't see a reason why it cannot be organized at the other places. I think it's not that cell is always going to shrink on one side, right? Sometimes it also has to form the opposite.
Because the example you showed where you had this linear and then it forms off a bend and then that forms a circle separately. That would require some cells on top to change.
Exactly. Yeah. And that's why I said that basically this has to be regulated in time and space. And when I say space, it's also the intracellular space. Sometimes, especially when the cells divide, you also have this contractile apparatus actually in the middle of the cell. And that's how you actually make the two cells separate. That provides the force for the separation for these two cells. Right. Right. So how do you test if the mechanical properties of this epithelium are altered? So we came up with two different assays. One is that we inject oil in the brain ventricle. So there is space in the zebrafish brain. That's what's called ventricle, and you can inject the oil, and you can let this oil sit there for some time. And so now you can imagine that this tissue is going to have a continuous stretch that will experience. And then you ask whether this tissue is going to deform over time. That's the first assay. And the second assay is that you keep on injecting the oil until it fractures. Right, so we use these two assays, and this amount of oil is good enough to induce enough tension on the tissue. We know that because the tissue reacts to this tension by increasing the cell proliferation, and in collaboration with Koti, we have also shown that using AFM that the tension is indeed higher when you inject this oil. So, and then you ask as I said, you inject the oil and do these two assays and ask what happens. So we basically saw two different outcomes when we use two different ways to inhibit cell proliferation. One is hydroxyurea treatment, and one is this morpholino, which will knock down the function of this particular protein. You see that here this oil basically kind of is projecting out this oil bubble, indicating that the overlying epidermis is deformed here, while it's not happening in this particular situation. On the other hand, if you do the fracture assay, it doesn't matter whether you inhibit proliferation by using hydroxyurea or NP63, the epidermis fractures much readily here. So when we do the fracture assay, we actually measure the amount of oil that you need to inject before the fracture. And you will see that the amount of oil required for fracture is much less. This indicates that the fracture strength is reduced independent of the condition that you use. While here we were not sure what was going on, eventually we figured out that this particular knockdown also results in an effect on the cell-to-cell adhesions, which is mediated by a protein called as equidarin. So these are the proteins which basically connect the two cells together. And we noticed that in this particular condition, the amount of this protein is much less. So one can do the genetic trick wherein you supplement now these animals with equidarin. And when you do that, these phenotypes get alleviated. So the bulge is not as much as it is otherwise in the absence of equidarin. Now, while this was true, if you inject equidarin, there was no consequence for the fracture phenotype. So even if you inject the equidarin, which is going to now strengthen the adhesion between the cells, they fractured more or less the same way as the non-supplemented animals. What it means is that this is something which is independent of adhesion. So what it means is that it is cell size which is driving this fracture, not the adhesion. That's the conclusion of this phenotype of these set of experiments. Then what is it that is actually linked with the cell size is the question that we asked. And we looked at a protein which is called ezrin. So the ezrin, what it does is that the actin cytoskeleton that we have been discussing, it connects this actin cytoskeleton to the apical membrane. So we looked at this protein, what is the status of this protein? And we realized that in the control embryos, when the cells are actually large, so during normal development, cells are to begin with are slightly larger, and then they become small. And as they become small, the you see that enhancement of the levels of these ezrin. But if you now inhibit the proliferation, this doesn't happen, right? The levels of the ezrin remain really low, whether it is a sham control or whether it's the animal which is injected with this large amount of oil. Now the question is, these are the quantifications. I'm not going to get into the details. The question is this just a correlation, or indeed this protein is somehow doing something in these cells? So how do you test that? So one way to test that is that you take this condition wherein the cells are large and you upregulate the levels of ezrin. So you now inject the RNA for this particular protein. So this more protein will be made and you will see increased localization. And then you ask whether the fracture strength now is better for such animals. Although now the cells are larger, you will have more of this protein and then the fracture strength will be restored. And these are the kind of experiments that we did. So this is the condition wherein the cells are larger. And we have now injected this molecule exogenously. And one way to test this is that you actually see the fluorescence because it is conjugated with green fluorescent protein. So this is the protein of interest. It is labeled or tagged with this fluorescent protein so we can see this. And if you now do the fracture analysis on these animals, which is done here, so you see that the fracture strength is much better as compared to the control animals. What it tells you is that this protein indeed is, rather its levels are not just correlated, but are causal to this phenotype. I'm not going to get into some of these details. So I'm going to give you the take-home message, which is basically now published. So if you look at these cells, and let's look at how the forces are operating here in these outer cells. So you're looking at these cells from the top here, and this is the section of the two cells. This is one cell, and this is the second cell. So these cells are connected with each other by echidherin, as I said. They are joined by these proteins, which are transmembrane proteins. I also told you about this actomyosin contractile apparatus, which is present at the cortex. Now, there is the tendency of this contractile apparatus is to constrict inwards, right? That's what it does. And when that happens, of course, cells are going to kind of go inwards. But what the adhesion does is it counterbalances this contractile force. So the adhesive forces are in the opposite direction to that of the contractile forces, which are the red arrows here. And these magenta arrows are basically the adhesive forces. Now, what we think is that these green arrows, which basically come from the ezrin, which is present on the apical side, they will also antagonize these contractile forces. So now imagine if you don't have these ezrin, when the cells become big, the ezrin levels go down. So these contractile forces or the effective contractile forces are going to be larger, right? So each cell will now show the inward constriction of some sort. Now imagine each unit constricting. So this builds up the stress in the epithelium. And now if you challenge this with something like the oil injection, which is going to apply tension, this falls much readily when the cells are bigger. So that's the model that we proposed. As I said, this is already published.
Just now mentioned is contrary to the earlier claim that ezrin is inversely correlated to cell size, right?
Yeah.
The less ezrin you have, the more size you say.
Yeah, when the cells are larger, then the ezrin is lower. Exactly.
Yeah, and so if you don't have ezrin here, the contractile forces dominate.
Yeah.
So it would be a smaller cell.
No, but it's also adhering to the neighboring cells with the echidherin. Okay, so now imagine the echidherin is trying to pull, trying to stay connected with the neighboring cells. Now it's one each unit, right? It's trying to kind of constrict inwards, you're right, but it's also connected with each other by the adhesion, right, by echidherin, right? And now you can imagine the stress on the junctions when it's constricting inward, right? And now if you challenge this with additional tension, it is going to fall apart easily. Does that make sense? Yeah.
Sure.
Yeah. Yes. Yeah.
Thanks. No, sorry. So this again, when you described the whole thing and you said that the protein which actually increases adhesion doesn't have any effect on the breaking, at the point at which it would break. I imagine, therefore, that it has to rupture along the cytoplasm and rather than between the cell boundaries, because otherwise I couldn't understand why that protein makes no difference.
That's true. I mean, we could never really figure out where the rupture happens. We don't have that possibility of figuring out, but that possibility does exist. What I would actually say, I didn't want to get into the details, but since you asked this, now, it's Kedirian. Yeah. Okay, so if you look at this condition here, now these are the wild type cells, which are normal size cells. Now, if you enhance the adhesion, this is now you're injecting equidirin. Now you have better adhesion. These cells actually now these animals have better fracture strength, right? So here, of course, the ezrin levels are also higher because the cells are smaller, and you have higher equidirin. So it works much better. So if one of the two are actually reduced or lower, then you are going to have these kind of fracture effects. Here, if you're supplementing more equidirin, the ezrin levels are low, and therefore the fracture happens readily, although you have better adhesion, right? But it's quite likely that the cell might also be fracturing in the middle, which we haven't really tested. So that's something we tried doing it, but some of these experiments didn't really yield us very conclusive results. Right. So I'm going to talk a little bit now about these projections that I was discussing earlier. So these are the projections that are formed again by F-actin. These are not just present on the fish surface, but they are present all over from fish to mammals, including in humans. So if you look at your corneal cells, you will have them, or the buccal mucosa cells or cervical epithelium. Many of these epithelial cells have these apical projections, which grow laterally long. And they have not been investigated in the past. Of course, people have documented their presence in a variety of different squamous epithelial systems. The research on them was quite, rare, or is not as deep, simply because it is not possible to have these projections formed on the cells which are cultured in a dish, right? So you need a tissue, or you need an organism that's growing so that you can look at these projections. And zebrafish became a very, very, very good model. And in fact, some of the early studies which we published were some of the first studies in this field in the context of how these projections are regulated. So what are these projections good for? They are important for retention of mucus. And mucus itself is a barrier that protects the underlying epithelial cells. And here we have imaged the fish epidermis, wherein we tried keeping the glycan layer or the mucus layer intact. And you can see the mucus layer, which is actually coating the microridges or the apical surface. And this is something which was done here in our SEM facility with Bhagyashri. I don't know if she's here. She helped us doing some of these analysis. So as I said, largely understudied, and so we had that as motivation, why and how these long structures form. But there was another big motivation, and that's how the ridges become long. Now, the other actin projections that people have been studying are microvilli or stereocilia, which grow finger-like, right? So these are finger-like projections on the apical surfaces. And what one needs to understand is that the actin actually grows against the membrane, right? So one can imagine if the actin grows against the membrane, you can deform the membrane and form a finger-like projection as long as you have enough scaffolding around it so that it doesn't buckle, right? But the question is, how do you actually form these projections which are laterally long? I mean, this is the cell biological question, which was quite intriguing. Look at this like a maze-like pattern. And how do these actin things grow laterally long was the question that intrigued us. And we have uncovered, sorry, I don't think we need to play this, the genetic regulation of this. So we have figured out multiple different components which are important for the elongation of these microridges. And here is one example. This is a mutant, APKC mutant, wherein you will see that the ridges have become extremely long. What it does is basically it prevents the localization of these two proteins, LGL and non-muscle myosin 2. These act as pro-elongation factors. So these factors are important for elongation of these ridges. And this prevents the elongation, the localization of, or rather not prevents, but it controls the localization of these two pro-elongation factors so that the ridges can grow up to a certain level, but not beyond that. And so there are, of course, many other factors that we have identified. And currently we are looking at another component which is called mTOR. Now, this mTOR is an interesting molecule. It responds to the metabolic status of the cells. What it means is that your epithelial surface can actually be modeled based on the, what is the metabolic status of the cell. So what we have shown is that if you inhibit the function of this molecule, you again see these ridges, their ridges become long. And this phenotype is very similar to the phenotype that we actually saw earlier with this when we have deficiency of this particular protein. There are two different kinds of complexes that this protein forms, and we have worked out the pathway by which this controls this TOR-C2 basically operates via phosphorylation of APKC in controlling the microridge elongation. And this is something I'm going to show you just one quick evidence how we tested this. In the APKC deficient background, you can inject the normal APKC or the plasmid that encodes for APKC and ask whether it rescues the phenotype. And now you also inject a mutant version of it, which will not be responsive to this activity of this protein, and ask whether that also rescues. And quickly, I will not get into details. Of course, the normal protein, when you have it present, now it's present only in these 3 cells, while the surrounding cells are still mutant. You will see that the ridges are now shorter. The same is true for this one here. But if you have a defective form of APKC, which now doesn't crosstalk with the TOR protein, you don't see this rescue. It tells us that these two are genetically coupled and are acting in a pathway. Now, yeah.
Could you explain the plot you're using as the next? Yeah, those shapes that it looks like.
The triangle and non-regular polygon put together. No, it's not a triangle. It's both non-regular polygons. So the green thing that you're showing.
Sorry, these are basically the quantification. This is the length that is plotted here. Sorry, I was pointing out to these patterns. So if you look at these patterns, this is basically quantification of how long these ridges are.
Yes, but what does that shape mean? Why is like the green, why is the bottom one longer on the right than on the left, for example? I'm sorry, the the green and the light green.
Yeah. Yeah.
Yeah.
What is what?
It's like a box.
It's a box, yeah.
Just to explain what that's like. How do you interpret a plot like that?
Yeah, it's very what like there is an x-axis, there's a y, but
Yeah,
what is
so
that shape?
basically this is the microridge length and this is in micrometers, and basically this depicts the entire spread of all these microridge lengths. And these are basically, this is an interquartile range, and we can discuss this together. And basically we compare these distributions. And what it tells us is that this distribution is not significantly different. Basically,
So the width along the horizontal of that shape corresponds to the width of some spread at that point in the length or something?
No, I don't think so.
Sorry.
But we can discuss the details. I also don't right away remember this, but we can discuss this. Right. So what is more interesting actually is, and which is, although it's not a story in a way, but it's something that we are taking it ahead. And this is where biologists will have to talk to people who are working beyond biology, like physicists and theorists. Is this question, right? These microridges actually form a variety of different patterns on the cell surfaces. And if you look at these are different fishes showing different kind of patterns. Even within a fish, if you look at the different parts of the body, the patterns change. Now, the question is, how do these different kind of pattern form? Of course, one way to do this is that you have regional differences in the signaling mechanisms that we discussed. The genes and the expressions are different. But we are asking if the curvature or if the tension which is present in the tissue, which is whether that is different and whether that is the reason why we have now different patterns that are forming. And to test this, we are now coming up with different kind of assays. This is one thing that we've started doing. We basically inject this oil in the brain ventricle, which expands the overlying epidermis. The ridges break down rapidly, but they recover over time. And if you now look at these two patterns, they look quite different. So this is the sham control. You see that the ridges are much densely packed when they are growing under tension, right? That's one thing, observation that we have. The other thing is that, of course, you need ways to quantify the patterns. You have to have the ways, yeah.
So in the previous slide, the image that you showed, is that the test condition, is it chronic or acute?
So no, this is chronic. Sorry, if it breaks, then it basically you will have a wound, so you can't really look at it. Right. Of course, you have a ways to figure out. So Clyde, who was a PhD student, he basically modified the algorithm that is used to assess the fingerprints. And he modified that algorithm and made it suited for our purpose. Now you can get a lot of information from these patterns, the number of ridges, how long they are, how many bifurcations exist, etc., etc. So that's one thing that we have done. And there was a postdoc in the lab who was also trying to set up the neural network-based approach to find out if the patterns can be classified based on how they are, whether they're long, complicated, convoluted, whether they're more simpler. And there is basically, if you take the head cell and the cell on the flank, it is possible to classify such patterns using the neural network approach. So that's something that we have now established. And what we are doing is that we are coming up with this ways to image these cells as they grow, as the microridges grow long. So these are the cells here, and these are the actin punctae. What we have done is that we have used a chemical which will break down ridges, and there'll be actin punctae. And Now we wash off this duct so that these ridges grow back again. They become long. And you will see this in the movie as they grow long. At some point, you will start that the ridges start to, especially in this cell, if you look, time, it's more obvious that there's still no ridges here. You start see, they're growing now, also here. And the way they grow is that there is a lot of fission and fusion that happens, but eventually, effectively, they grow long. Now, what this fission and fusion is good for, we don't understand. But what we are trying to do now is we're trying to extract the information in the sense how many fissions happen, how many fusions happen. Is it that now the fusions are more at the later time points, and all those quantitative aspects we are trying to get from such movies. It's of the filaments of these dots. Right. So that's one thing that we're doing. We are also trying to measure the height of these projections. So it was until now we have been looking at all these projections in 2D. You look at them from the top, but they have heights, and this is where Koti's expertise was quite useful to us. So in his collaboration, we have actually been able to figure out how tall these are. Of course, we knew this from the electron microscopy studies, but those are quite cumbersome techniques. And Koti has much, I wouldn't say much easier, but relatively easier way to look at this. Of course, there were teething issues, which Koti and Satya, who is also in the audience, who sorted out. And now this gives us the additional parameter. And what it seems like is that when the ridges are short punctate, they are not really tall. And so if this is the situation wherein the ridges are extremely small, when we have just washed off the drug, but they have not grown long, and they are as tall as maybe 70 nanometer, these small membrane deformations. But as they grow long, they also grow tall. So somehow the length and the height seem to be linked. Now, this is something that we are talking to Madan and other theorists to figure out how these things are kind of, growing laterally long. So with that, I think this is my last slide. So what we have shown in the past is that how the polarity modules and actin myosin contractility is important for formation of these ridges. The TOR signaling now allows us to ask whether the metabolic cues are going to impinge on formation of these, and if there are mechanical cues which are, feeding into the polarity modules to give such patterns. And this is something I want to finish in next 7 years before I retire. That is 7 years is my time. Right. And something that we started, so we are now going beyond developmental regulation. During development, of course, these programs are genetically hardwired, but many of these epithelial tissues are still functioning in our bodies, your gut, your lungs, they come in contact with a variety of different conditions. For example, we are asking how hypoxia would influence the epithelial architecture, how the nutrition would play a role in defining the gut architecture, for example. And of course, the physical forces, how they are going to play a role. And in the context of nutrition, we are also looking at the gut and how the gut morphology, so this is mammalian gut here, how the mammalian gut would respond to the nutrients and nutritional composition in general. And this is something that we have started looking at very recently, and something that we are doing as a part of this collaborative project. We call it Arumda. It's a big consortium of variety of different labs, which is being kind of coordinated here in TIFR Mumbai and in TIFR Hyderabad by me and Ullas. And this is something I hope I'll give one more colloquium before I retire again, and hopefully then we will have more information on these things. right? People who have helped us a lot in-house, we have been helped by Koti's lab a lot, and Ravi, who have been helping us with modeling the APKC structure and how the phosphorylation influences that. And this is the current group and of course the past people. Thank you for your attention.
Thank you, Mahendra. Seem to have generated some interest, judging by the questions. I learned a few things about epithelial morphogenesis. Questions? I'd like to first.
Very nice talk, Professor. I had a question that when you are explaining about the mechanism of the fracture of this epithelial cell membranes, can this surface tension play a role in the mechanism of the fracture or anything? Like
surface tension of what? Of the
of
oil?
the cell, basically, that if it has larger surface, it will have larger surface tension. So that means that it will be more prone to breakage or something like that. And if it is smaller in its surface, then the surface tension is low, so it has lesser tendency to break. I mean, I was just thinking.
Okay, now that you made me think about it, so we will think and maybe we can chat over a cup of tea.
Okay, thank you.
It's an interesting thought. Yeah.
Just, you showed us the sort of nice change in the gut morphology in the MYO5 mutant, where it did not have those folds. I'm just wondering if that results from the same cellular phenotype that you see in the epidermis. I mean, what is the reason for not having folds? Do you know if cell numbers or cell size or something else is affected in those mutants? The diameter of the gut is affected in any way?
Yeah, so the cell shape is affected in the myosin mutant. So typically when the folds form, the cells actually expand their apical surfaces, especially those that are at the apex of the fold, which is important. Otherwise, you cannot kind of accommodate the entire surface, and that doesn't happen in this mutant. But of course, it's not clear whether that is the sole reason. The proliferation does not seem to be affected in these mutants, but I'll go back and check. But we could not really figure out exact reason why the folds don't form. And as you probably know this, that the fold formation also kind of is linked with the surrounding muscle layer and the confinement of the muscle. So the growth of the epithelium, when it happens in a confined environment, then it leads to these formation of the folds. We haven't looked at the muscles which are surrounding the epithelium, whether there is any defect there. So that's something which is not clear. Yeah.
Next question.
Hi. Yeah, very nice talk. So I have this question comparing epithelial cellular dynamics, which you have been working on, versus wound healing. Here, wound healing. So here for the epithelium, it might be natural. So that maybe evolution has really figured it out how to do this. But in the case of wound healing, it's basically an unanticipated thing. Do you think basically the cellular size, dynamics, etc., where both chemical cues versus the mechanical cues, which both play important roles? Are there any common mechanisms by which both might be operating, or they are completely different mechanisms by which the mechanical aspects actually come into play?
Okay, mechanical aspect, I'm not so sure. I mean, it will have an influence that in wound healing, especially, it doesn't, in vivo context, especially, nothing comes to my mind right now. For that, you basically have to make a wound and apply tension. That's what basically one has to do. And I don't know if that has been, I think in cell culture that has been done. And of course, it will have a consequence. But I mean, it's obvious. Probably both are going to play a role. The tension as well as. So what something that quickly comes to my mind is that if you actually stretch the epithelium, the proliferation rate probably has to be higher than the normal condition. And of course, the cells also have to migrate towards. So I would imagine the proliferation rate would be different, and the motility of the cells probably will be slightly different. The time scales would probably be different. But I'll go back and check, and we can talk about this.
Yeah. And I had a similar question to the earlier one, which was, as the cells grow bigger, the ratio of their surface area to their perimeter will change, so that must be playing a role, right? And even if I have the same amount of adhesive protein per unit length along the perimeter, the mere fact that you're subject now to more force because the surface area has gone up, when you inject your oil or whatever, those things necessarily would play a role also, right? In how quickly they're ruptured.
They will, of course.
So I was wondering whether you're normalized for that sort of thing or factored it in some way, or.
No, I mean, we basically went the Ezrin way right now, but we haven't looked at the other physical factors. We did some theoretical analysis with Mandar and Amdar, and I did not get into the details of that. But I don't think we really scored for these. Rather, we factored in some of these aspects.
So I had one small doubt. When we are talking about proliferation here, are we talking about cell size or cell number or both? Like when we said that the proliferation was inhibited in one of the slides that you mentioned, are we inhibiting the cell division or are we inhibiting actin polymerization?
No, we are inhibiting cell proliferation. And when you inhibit cell proliferation, because the number will go down and the cells have to then become big. I think that's how it is.
Last question.
I mean, of course, this is during the growth. I mean, if it's a tissue which is not growing, then it will not have a consequence.
So these things you discussed for epithelial cells, is it a similar mechanism also for like muscle cells? They also have to deform, but is it using a similar contractile apparatus?
Say it again.
The cells in your muscles which need to deform much more.
Of course. Yes, you have actin and myosin, exactly. Is this the organization of actin and myosin is different? It is basically organized in some kind of blocks, the pattern blocks.
Okay.
So that you have more directional contractile apparatus, right? Which rather than this is slightly different. There it is more, they are organized in bands so that you get the constriction.
Okay, maybe you'll say let's chat over chai, but I'm intrigued by your conversation with Mustansir about hexagons and convexity and so on, because these cells are actually three-dimensional, right? And the z-axis we are not looking at here. And isn't convexity and so on induced by a shrinkage of the lower surface area so that the convexity is not.
No, but you need to have all the cells identical. They all have to be hexagonal, which is not the case here. So we have a distribution of.
No, but I mean, in terms of convexity, it's the z-axis that we are not looking at, right? The lower hexagon being smaller than the upper hexagon of a cell will induce convexity very easily.
You mean because it is bilayered? Is that what you're saying?
I mean, the cell has a thickness, right? And
you're talking about individual cell having
and
height.
in section, you often show how the lower surface is much tinier than the upper. So we don't need to invoke polygonal mechanisms for convexity because of the cell's depth, and that the different surfaces are modulated differently, no?
Yeah, but I mean, this depth, I mean, if you really look at it, I mean, I don't know, I'm trying to imagine this, but if you have, these are extremely flat cells in some sense. I mean, in principle, you can imagine these as really as
They don't have as much depth as some of the
they
other
are not
cells
really
you
tall. These cells
mentioned.
especially they're not as tall.
Okay, okay.
Yeah.
Let's thank Mahendra again for the talk. So please join us at the West Canteen for the tea.